Hello to the seekers of knowledge. Welcome to Uniway's Academy of Graduate Studies. We are here to inform you the beauty of biochemistry. All of our team try to do their best to upgrade your knowledge about sciences, and I hope it will be worked in this channel. Now today, join us with another section. In this video, we're going to talk about buffering system in human bodies and different types of it. Be with us till the end of the video and don't forget to like it and write your opinion in the comment section down below. And of course, subscribe to our channel. First of all, let's take a look at the reason why our body needs this system. We know that the muscles in the body need fuel to work. This fuel is a chemical called adenosine triphosphate or ATP. The body cells only store enough ATP to last about 3 seconds, so systems in the body produce it when it is required. As you see, this molecule consists of 3 phosphate group, 1 ribose sugar, and 1 nitrogenous base, which is adenine. There are 3 main systems that produce cellular ATP, which is involved in generating energy for cellular processes. Number 1, phosphagen system, a high energy compound in the muscles called keratin phosphate donates a phosphate that can combine with ADP to form ATP very quickly. This reaction is catalyzed by keratin kinase. This supply of keratin phosphate only lasts about 8 to 10 seconds, so it's useful for quick sprints. Number 2, glycogen-lactate system, which is anaerobic glycolysis, is the transformation of glucose to lactate when limited amounts of oxygen are available. Anaerobic glycolysis is an effective means of energy production only during short, intense exercise, providing energy for a period ranging from 10 seconds to 2 minutes. ATP is produced from glucose before the cardiovascular system has responded to change in movement. Pyruvic acid, which is the final product of glycolysis, stays in cytosol due to the lack of oxygen and then turns to lactic acid. Increase of lactic acid causes muscle cramps and pain. Number 3, aerobic system or respiration, which involves 4 stages. Glycolysis, a transition reaction that forms acetyl coenzyme A, the citric acid or Krebs cycle, and an electron transport chain. Respiration is a series of chemical reactions, but the equation summarizes the overall process. Aerobic respiration breaks down glucose and combines the broken down products with oxygen, making water and carbon dioxide. The carbon dioxide is a waste product of aerobic respiration because cells do not need it. Now you may ask, what's its problem then? Well, energy required for muscle contraction in the body comes from hydrolysis of ATP. When a molecule of ATP is hydrolyzed, 1 phosphate breaks off to produce adenosine diphosphate or ADP, a free phosphate ion, a hydrogen ion, and a large amount of energy. No matter which system the body uses to produce ATP, hydrolysis of ATP to ADP produces excess hydrogen ions. We know that the body must maintain a stable pH in order to function properly. So this is when buffering systems do their job. What is buffering system indeed? pH of blood and extracellular fluids is in the range of 7.35 to 7.45. This range is biologically important, so it should be maintained. Kidneys and lungs are two organs of the body that regulates pH accompanied by buffer systems. As it said in the last YouTube video, buffers are a solution containing weak acid or weak base which resist change in pH by absorbing or releasing hydrogen ions. There are three main buffering systems in the human bodies. Number 1, the carbonate-bicarbonate buffer. Number 2, the phosphate buffer. And number 3, the buffering of plasma proteins. After about 2 minutes of moderate exercise, the cardiorespiratory system is able to deliver sufficient oxygen to working muscles and get rid of the carbon dioxide that is produced. The products of aerobic metabolism, water and carbon dioxide, react together to form carbonic acid that dissociates into hydrogen ions and hydrogen carbonates. It's the most important buffer of blood. Carbonic anhydrase enzyme catalyzes this process in red blood cells, walls of pulmonary alveoli, and epithelial cells of kidney. Zinc element is a cofactor which is essential for carbonic anhydrase function. This buffer contains weak acid, carbonic acid, and its conjugate base, bicarbonate, so that any excess acid or base introduced to the system is neutralized. As it's shown, carbon dioxide reacts with water to form carbonic acid, which in turn rapidly dissociates to form a bicarbonate ion and a hydrogen ion. Now let's explain its function based on Henderson-hasselbalch equation. pK value of carbonic acid is equal to 6.1. The concentration of carbonic acid is equal to KHCO2 multiplied by the pressure of CO2. KHCO2 refers to the solubility of carbon dioxide in blood, which is approximately 0.03 millimole per liter. So in this case, we have this formula. In human body, normal pH is 7.4, which means the ratio in parentheses should be around 20. This ratio should never be changed, and this is the responsibility of the carbonate-bicarbonate buffer. In tissue, cellular respiration produces carbon dioxide as a waste product. As one of the primary roles of the cardiovascular system, most of the CO2 is rapidly removed from the tissues by its hydration to bicarbonate ion. The bicarbonate ion present in the blood plasma is transported to the lungs, where it is dehydrated back into CO2 and released during exhalation. These hydration and dehydration conversions of CO2 and carbonic acid, which are normally very slow, are facilitated by carbonic anhydrase in both the blood and duodenum. While in the blood, bicarbonate ion serves to neutralize acid introduced to the blood through other metabolic processes, for example lactic acid and ketone bodies. Likewise, any bases, for example urea from the catabolism of proteins, are neutralized by carbonic acid. The bicarbonate reabsorption in the proximal tubule of kidney was mediated by hydrogen ion secretion, but that carbonic anhydrase located in the luminal membrane of the cell prevented carbonic acid from accumulating in the tubular fluid. The bicarbonate ion reabsorption in the renal tubules plays a critically important role in maintaining the global acid-base balance. Loss of bicarbonate ion causes metabolic acidosis. Proximal renal tubule is the major site for bicarbonate reabsorption, accounting for more than 80% of total bicarbonate reabsorption along the nephron. Over the past more than half centuries, tremendous progresses have been made on understanding the molecular mechanisms underlying the bicarbonate ion reabsorption in proximal tubules. The transepithelial movement of bicarbonate involves the coordinated operation of mechanisms on both the apical and the basolateral membranes of the epithelial cells. This system is the most active one in intracellular fluids, especially in kidneys. A phosphate buffer depends upon two principal components: the weak acid monosodium phosphate and its conjugate base, disodium phosphate. If a strong acid is added to this solution, hydrogen ions will be accepted by the hydrogen phosphate base, forming dihydrogen phosphate. Phosphates have a very high buffering capacity and are highly soluble in water. Whenever hydrochloric acid, which is a strong acid, is added, this buffering system converts that to a weak acid, such as monosodium phosphate, which its ionization is too lower than HCl's, so it avoids drastic change of pH level. However, this buffer has a number of potential disadvantages. Phosphates inhibit many enzymatic reactions and procedures that are the foundation of molecular cloning, including cleavage of DNA by many restriction enzymes, ligation of DNA, and bacterial transformation. Because phosphates precipitate in ethanol, it is not possible to precipitate DNA and RNA from buffers that contain significant quantities of phosphate ions. And of course, phosphate sequester divalent cations such as calcium and magnesium. If we want to explain the function of this system based on henderson-hasselbalch equation, we should know that pK value of phosphate is 6.8. In human body, normal pH is 7.4, which means the ratio in parentheses should be around 4. This ratio would never be changed, and this is the responsibility of the phosphate buffer. All proteins have both amine and carboxyl group, so they can show buffering specialty, because they act as a receiver and donor of hydrogen ion. This is the most abundant buffering system of the body. Protein buffering is a description of the process wherein proteins compounds consume small amounts of acid or bases. For example, hemoglobin is a protein that binds to small amounts of acids in the blood, removing the acid before it changes the blood's pH. There is an imidazole ring in histidine's structure, and its pK is very close to physiological blood pH. Actually, histidine is the only amino acid that can be a buffer in physiological pH. Hemoglobin has 38 residues in the structure, which means that it could act as a buffer in red blood cells. Hemoglobin is the principal protein inside of red blood cells and accounts for 1/3 of the mass of the cell. During the conversion of CO2 into bicarbonate, hydrogen ions liberated in the reaction are buffered by hemoglobin, which is reduced by the dissociation of oxygen. This buffering helps maintain normal pH. The process is reserved in the pulmonary capillaries to reform CO2, which then can diffuse into the air sacs to be exhaled into the atmosphere. Now let's talk about albumin protein. This protein, of course, is a buffer in plasma because it has 18 histidine residues in its structure. Albumin is the most abundant plasma protein in blood, and it constitutes 50% of protein in blood. This protein is made by hepatocytes, and it maintains intravascular onconic pressure. Of course, albumin is the most important carrier in blood, which carries hormones, ions, and medications. Buffering capacity is defined as the soil's capacity to maintain a relatively stable pH despite the presence of acidifying or alkalizing factors. Soil buffering capacity is caused by the protonation of minerals and organic material that occurs in the soil or is intentionally added to the soil. A higher buffer concentration has a greater buffer capacity. This means that a greater amount of hydrogen ions or a stronger acid would have to be added to disrupt the equilibrium and change the pH of the buffer. Buffer capacity is also affected by the relative concentration of the buffer components. That's it, guys. Thanks for watching. Hope you enjoy this video. You can contact us on social media. We would appreciate that if you could write your ideas on the comments section. Thank you again. Goodbye.